Unit 3.1c, we're going to start out talking about how we sequence genomes. So we live in a remarkable age. We have incredible resources and technology that allow us to peer in at the molecular level at the genomes of organisms. The human genome was sequenced in 2001. This genome sequencing project has been likened to putting a man on the moon, in both in the level of complexity and the amount of time and effort it took. So it actually took about a decade to complete and untold billions of dollars. I don't even really know for sure because there are lots of different people working on it, different organizations. Today that has remarkably improved. We can now sequence an entire human genome for just a few $1,000s and have it done in a couple weeks. A lot of the reason it took so long to sequence was not only did we need to did we have outdated technologies, slower technologies, but also we had to develop them. So some of the things we didn't even know how to do or how to handle the large amounts of data and the algorithms needed to assemble all of the data coming off. But even though we can do it much, much faster today, the way that we sequence genomes is still roughly the same as it was from the original. We don't have a better way to do it. So you can't just take a long string of DNA and read the sequence off A's, T's, C's, and G's from one end to the other. If you could figure out how to do that, anyone could. They'd win the Nobel Prize in chemistry, and because there isn't one in biology, I don't know why, but they'd win the Nobel Prize, they would win, make all sorts of money, but we can't do it. But the way that we are stuck with doing it, we've gotten much, much more effective and efficient at doing it. So this is whole genome sequencing, and it's still described as a shotgun sequencing process. And you'll see the reason we call it shotgun sequencing. So you need to know these steps. In fact, let's type them in here as we go. And again, if you want to read them and look at them, that's great. But even better, write them down. Just take some notes. Again, sometimes the very act of just writing those down will help you. So let's put them in here. If we were in class, I might do this on the overhead, but we can put it here. Okay, so number one, when we're going to do shotgun sequencing, the first thing we need to do is get lots of copies. Now, for some organisms, that's fairly easy. You can take bacteria and you can grow them till you have millions and millions of individuals. You can get humans and take a large tissue sample, liver sample that has hundreds or thousands of cells in it. So it's not too tricky. You can do this with most organisms. Some organisms, it's a little bit harder, right? We might have a little bit of trouble getting the number of organisms we need. Let's get this just a little bit smaller here. I want to go smaller. There we go. Okay, so there are chemical ways that we can get these lots of copies. We could say, we could say, hey, let's use a PCR or some sort of a amplification procedure to make extra copies. Okay, so once we have those lots of copies, we break them into small pieces. Okay. Now, we do this chemically, so they all end up about the same size. There are a couple ways you can filter out pieces that are too big or too small, but you end up with billions and billions of small pieces of DNA that represent maybe hundreds of thousands of copies of the original genome. And they're all identical. The copies are identical, so we have many of these pieces that overlap. And then, once we do that, we can sequence these small pieces and then assemble them together. And all the assembly is done on a computer, right? So we're not physically taking DNA and pasting it and putting it together, but we do it on a computer, and we do it in a way that is usually parallel, so using many, many processors and lots of memory, but we now have computers that can do this rather quickly and rapidly, okay? So those are the three, let me grab this here. Those are the three basic steps. You need to know those, okay? And so once we have the genome assembled, then we can begin to describe it, annotate it, map it. We talked about that a little bit in 3.1b, right? So we have human genome in 2001. We had the fruit fly genome, which was smaller, so we were able to do it first. Notice it's quite a bit smaller. And so as these genomes started rolling in, people said, oh, this is cool. Let's start studying them. Let's start figuring out. One of the very first things they did, kind of simple, I said, how big are they? And there was this fairly straightforward idea, hypothesis as people did this, that big complex organisms like mammals, humans, right, would have big genomes. And simple organisms, fruit flies maybe, or maybe even more simple like this, would have smaller genomes. And with our first samplings, that seemed to roughly hold true. So notice that the worm, the roundworm here, C. elegans, is rather simple. It has only 100 megabases, 100 million base pairs in its genome. The fruit fly has a little bit more. That's a little bit of a surprise that the fruit fly is much more complicated than C. elegans. And then humans are much larger. But here's a plant, right? And so when you have a small sample like this, it's really hard to know. And so as we start getting more and more and more genomes, we realized that this early hypothesis didn't hold up. Here's just one extreme example. And I've chosen this for effect, but it illustrates it. Here's an amoeba, single-celled. It's eukaryotic organism, so it's got a nucleus and organelles. But it's very simple, right? Not very complex. We don't have tissue types. We don't have cell-to-cell communication. It's much simpler than the cow, okay? Yet, this amoeba has 670 gigabases in its genome. That's a billion base pairs. So 670 billion base pairs, close to a trillion. The cow only has about 3 billion. By the way, about the same size as yours and mine. About 3 billion base pairs in its genome. Still a large genome, but 200 times smaller than this simple amoeba. And so as these genomes started rolling in, we started just identifying their size at first, we came up with this idea that we cannot predict organismal complexity based on genome size, and vice versa. Looking at genome size, you can't tell how complex an organism is. So this is known as the C-value paradox. There is no correlation between complexity and genome size. And you can see it here in this graph. And notice that some groups of organisms like lungfish and salamanders have notorious large genomes, but they're much simpler than all mammals, which all mammals have smaller genomes. And there's long overlap. So the protozoa, which the amoeba falls into, has this like 5-fold almost. Notice this is a logarithmic graph, so every gradation in the graph that we move up, we've basically gone in 10. So this would be, to here would be 10 times, 100 times, 1,000 times, 10,000 times. So maybe 50,000 times bigger genome of the amoeba than the smallest protozoan. So incredible range and no clear correlation between complexity. Now notice if we look at the prokaryotes, although not 100%, prokaryotes do generally tend to have smaller genomes than eukaryotes, although there is a little bit of overlap with the fungi, just barely, and with some of the protozoa. So just be aware of that. That's the C-value paradox, especially pronounced in the eukaryotes. So for many, many years, we didn't know why. Why is there so much DNA in an amoeba and so little, relatively little, in a cow? This was a big mystery. It was a paradox. But as we began to explore and annotate and map genomes, we found our answer. And it's actually a rather simple answer. And we'll look in a little bit more detail, but it's a simple explanation. The simple explanation for the C-value paradox is the non-coding DNA. So amoebas have way, way more non-coding DNA. Maybe 99.999% of their genome is non-coding, and the rest is genes. A cow still has lots of non-coding DNA, so it's not like, oh, it's 60% genes and only 40% non-coding. The cow is similar to you and I. About a little bit less than 2% of its genome is protein coding, so that leaves about 98, a little bit more than 98% that is non-protein coding, but still much less than the amoeba. So, C-value paradox, simple explanation is non-coding DNA. But then the question arises, why do we have so much non-coding DNA? What's going on here, right? So the non-coding DNA is the explanation for this plateau effect among the eukaryotes. So notice that among the viruses and the prokaryotes, there's close to 1-to-1. It starts to taper off a little bit, and then we just get this flat where there's no prediction between how big your genome is and how much coding DNA you have. There's really not a correlation. And that also goes to the same thing for complexity, okay? So why do we have so much non-coding DNA? And the answer is, in short, evolution, but it's a little bit complicated. And so there were some early hypotheses that maybe we needed large genomes to support large cell sizes. So as cells get bigger, and this was borne out by some observations like salamanders, which have large genomes, have much bigger cells on average than frogs. But that didn't really, that's correlation, not causation. Just because we have big cells doesn't mean you need genomes to support them. There might be other reasons and contributing factors to that. So this one really doesn't hold up. And so basically we're going to simplify it. This is what we're going to say. That the amount of non-coding DNA in your genome and your genome size in general is just simply based on a long process of adding DNA to your genome, mutations that add extra DNA, right? So we talked about those like a duplication event, right, where we have a chunk of a genome or a chromosome that's duplicated, or many insertion events, and your genomes would get bigger and bigger and bigger over time. We do have mutations that also remove DNA, and this occurs for prokaryotes, but also applies to eukaryotes also. And so we'll look about some of these factors in detail, especially in the eukaryotes, but we could lose a DNA segment, or we could gain a new gene from horizontal gene transfer for a bacteria, or a non-disjunction event, so you have an extra chromosome and you're carrying extra DNA. And so genomes generally get bigger and bigger and bigger. And many of the mutations that add DNA don't have a big phenotypic impact. So you got a little bit of extra DNA. But if you're starting out with, say, 1 million base pairs and you have mutations, so you have 10,000 extra, right? That's only like 1% of your genome. It's not a big deal. It doesn't take that much extra energy to copy it, so they're effectively neutral. And in fact, you get a few extra genes maybe, and maybe the extra copy of a gene might be a good thing. So by and large, we have this balancing effect where mutations bring in DNA, much of it non-coding, and mutations remove DNA. And if over time the mutations that bring in DNA are more common or kept more commonly than the mutations that remove DNA, genomes will get bigger and bigger and bigger. And so amongst the salamanders or those amoebas or lungfish, which have these notoriously big genomes, they've just had ancestors that have some sort of a mutational process that adds DNA. It's not really a good or a bad thing, it's just there. So genomes get bigger and bigger and bigger over time. And so we can see that that would not explain, it wouldn't make them more complex. It just means they're carrying around more DNA, and most of it's going to be non-coding DNA. Okay? So that's C-value paradox. Pretty simple and straightforward. That's it.